Today I will be going to discuss the immune system of fish, biological methods, disease diagnostic, case processing. Introduction. Vaccines stimulate the immune system to mount a defense against a pathogen. The development of effective vaccines against viruses, parasites, and intracellular pathogens. Vaccination by injection is the most reliable and effective system for vaccine delivery in fish, and the protection is by far the most effective and long-lasting. To be able to understand and discuss the different types of nano-delivery system tools and the difference of innate and adaptive in fish immune system, and to discuss the importance of biological methods, and to be able to present and discuss the role of disease and practices to prevent stress to the fish, to identify the different types of diagnostic tools of fish disease. Nano-delivery system tools targeting the fish immune system. Alginate. Alginate is found naturally in brown algae such as Laminaria hypoborea, Laminaria digitata, Macrocystis pyrifera, and Lesonia nigrescens. Alginate monocular phagocytic system can be produced by classical techniques such as air atomization, emulsification, gelation, and complexation with quaternary polymers, or by new methods such as spray drying, electrohydrodynamic impinging, aerosols, and inkjet or drying process that enable a better control of the size. Encapsulation of bacterial antigens in alginate microparticles. Formalin-killed bacteria have been widely used as antigens for the fish vaccination in some diseases, mainly caused by gram-negative bacteria in some diseases. In general, formalin-killed bacteria vaccines provide excellent levels of protection by itself or in combination with an adjuvant. The oral administration of formalin-killed bacteria encapsulated in alginate monocular phagocytic does not work very well, and only when combining the alginate monocular phagocytic with the formalin-killed bacteria vaccines they obtained a longer lasting protection. The oral administration of alginate formalin-killed bacteria from Lactococcus garvieae in rainbow trout provided by the low levels of protection against Lactococcus garvieae infection were in 35% relative percent survival at 30 days, compared with the naked formalin-killed bacteria. Vaccines administered intraperitoneally were in 100% relative percent survival at 30 days and 40% at 90 days. These results were improved when fish were immunized orally a second time with the alginate vaccine 3 months later in 61% relative percent survival at 180 days. Functionalization of carbon nanotubes. This process can be divided into two different approaches, which is covalent and non-covalent. Covalent are the attachment of different chemical groups such as sulfonate proteins, and bovine serum albumin has been used to design nanoparticles for fish. To evaluate immunotoxicity of functionalization of carbon nanotubes in multi-walled and single-walled of chemical groups such as sulfonate, sulfonic acid, and polyethylene glycol were tested for toxicity in head kidney macrophage isolated from omikis. The difference of single-walled and multi-walled of carbon nanotubes. Multi-walled carbon nanotubes containing anionic groups cause the highest stimulation, while the single-walled carbon nanotubes containing neutral groups cause the least reaction. Encapsulation of viral DNA in carbon nanotubes. To date, only one work has evaluated carbon nanotubes as a DNA delivery system in fish, but with promising results. were loaded with a plasmid encoding the protein of grass carp reovirus. Single-walled carbon nanotubes in grass carp was detected at high levels in muscles at 28 days post-injection. Intramuscular injection in wherein the treated fish showed good protection level even at low plasmid doses when compared with the naked plasmid. Liposomes. Liposomes are spherical closed structure composed of phospholipid bilayers which enclose part of the surrounding solvent into their interior, such as extrusion techniques is the most common method to prepare liposomes because it allows a better control of the size and the polydispersity index. The common technique method in liposomal formulation extrusion is a process in which micrometric liposomes are structurally modified to large unilamellar vesicles or nanoliposomes depending on the pore size of the filter used. Compare extrusions in microliposomes compared to macroliposomes. Nanoliposomes provide more surface area and have the potential to increase solubility, enhance bioavailability, improve controlled release, and enable precision targeting of the encapsulated materials to be greater extent. Encapsulation of viral antigens in liposomes. Immunostimulant-loaded liposomes are also developed to protect fish against bacterial and viral infection. For example, polyinosinic polycetidylic acid is a synthetic analog of double-stranded RNA and is a typically molecular pattern associated with viral infections. When combined with liposome, it is a strong stimulus to the innate immune system. Liposomes encapsulating both poly and liposomes elicited a pro-inflammatory and antiviral response. Zebrafish, hepatocytes, and throat macrophages. Biological methods to develop by agriculturists to control insect pests. The use of different species of carps to control the proliferation of water lilies and other plant pests that choke ponds and rivers. To minimize or control infestation or infection. Avoidance of pathogens. Under this condition, the fish or the cultured organism has to compete with each other for space and dissolved oxygen, which is they confronted with the stress of exposure to increased metabolites such as ammonia, carbon dioxide, and dissolved or suspended organic matter. Pathogens-free water supply. The most commonly employed methods for controlling the presence of these microorganisms are by sterilization of incoming seawater may control the number of potentially harmful microorganisms entering the aquaculture system but not necessarily totally eradicate these microorganisms in the water. Sterilization, filtration, ultraviolet light, ozone treatment, chlorination. Pathogen-free diets. Commercial feeds involve high temperatures of about 120 Celsius for short periods that should be enough to pasteurize, if not sterilize, the feed. The feeding of live food common problems. Specific pathogens free stock. The use of specific free stocks word stock is proving to be one effective managerial control measures, which minimize the likelihood of epizootics due to an identifiable pathogen. Disease infection and international regulation. Disease infection. A fish health inspection is a procedure by which a sample of fish collected from a defined fish population and examined for the presence of certain specific pathogens. International. Stocks that are pathogen-free are grown into broodstock in an approved quarantine facility where they are regularly examined for pathogen. The role of stress and disease. Stress is defined as physical, chemical, or biological factors that cause bodily reaction that may contribute to disease and death. Aquaculture practices that increase stress are high stocking densities and poor water quality. Injury during handling, chasing, netting, sorting, and shipping. Improper nutrition. Poor sanitation. Some managerial practices that may help prevent stress. Water quality. Maintain the appropriate or optimal pH, alkalinity, temperature, and salinity for the species. Handling and transporting. Minimize the number of times that fish are handled or lifted from water, and if possible use anesthetic to slightly sedate the fish. Nutrition. Use proper feeding rates of feeding schedules. Sanitation. Make sure that water supplies are not contaminated from the source. Diagnostic tools used in fish disease. Microscopic diagnosis. There are commonly used in fish disease diagnosis are light microscope, electron microscope. Light microscope. The light microscope is the most commonly used microscope in disease diagnosis. Electron microscope. They are broadly classified into two types, which is transmission electron microscopy, scanning electron microscopy. Transmission electron microscopy. They are used for observing the change in the tissue at higher magnification. Scanning electron microscopy. They are used for observing the surface level change in the tissues at higher magnification. Microbiological diagnosis. Histology is a branch of biology that involves the microscopic examination of thin, stained tissue section in order to study their structure and function, and, in the case of histopathology, to determine change which may be due to pathogens and disease. Bacterial diseases can be identified by various microbiology methods. The samples are taken from the fish under aseptic condition, and they are first grown in unselective media. Then the various microbial diagnostic methods are used. The different microbiological diagnostic methods are staining methods, motility tests, culturing in selective medium, biochemical tests. Immunological diagnosis. These tools are used for the qualitative and quantitative estimation of the pathogens and or the protective antibody. It is apparent that conventional diagnostic method rely solely on the microscopic examination in its visual recognition, which requires a great deal of experience with the organism that often change so dramatically in their morphology during the course of their development. Molecular diagnosis. Molecular techniques are potentially faster and more sensitive than cultures, serology, and histology methods that are traditionally used to identify fish pathogens. Pathogens can be detected from asymptomatic fish by molecular diagnostic techniques, so disease outbreak could be prevented.